today, I'm going to give you an overview of what hemorrhagic disease is. I'm going to cover the research problem that I was trying to help solve. I'm going to talk to you about the tools or the animal models that I used in my project, and then I'm going to go over, so it's like a general background, and then I'm going to go over the project specific objectives, our research methods and results, and then I'll give you some conclusions and future applications, and then we'll do the acknowledgements. So we're going to start off with hemorrhagic disease. So hemorrhagic disease is most commonly seen in white-tailed deer in the U.S. There are two causative agents, epizootic hemorrhagic disease virus, which is what I'm focusing on for this presentation, and then also blue tongue virus, which is shortened to BTV. And so there are three forms of the disease: acute, peracute, and chronic. And so when we see these in actual animals, we will see commonly that we find deer dead, and that's usually during the acute stage, and they're found near bodies of water. And then in the chronic stage, you'll see that they sometimes have hoof deformities, indicating that they were infected at one point. And then in sheep, which is more of a blue tongue, but also gives some really good characteristics of hemorrhaging, and then the swollen tongue, which is sometimes blue, so that's where you get the blue tongue from. So acute symptoms in deer will include swelling of the head and neck caused by edema due to leaky vessels. We'll also see respiratory distress, excessive blood-tinged salivation, and then the sudden death, as I was saying before. And so these are just some pictures of what the disease looks like in deer. And so you can see what they look like when they're still alive, and then we see what some of the hemorrhaging looks like in the organs during post-necropsy. So EHDV is found in various places across the country. It's commonly found. Its distribution spans a wide range. The serotypes that are native to the U.S. are EHDV1, EHDV2, and EHDV6. Prior to 2012, however, only serotypes 1 and 2 were present, and then in 2012, serotype 6 began circulating, and it was discovered to be a reassortment between EHDV2 and an Australian EHDV6. Outbreaks occur in the U.S. in the late summer or early fall. White-tailed deer are most commonly affected, but it can also affect other wild ruminants and occasionally domestic animals, usually cattle. When it does infect cattle, you don't see mortality per se, but you see morbidity or a decrease in milk yields. So this map is coverage of EHDV between 1980 and 2003. And then since 2003 and ongoing, so past 2013, we've seen a northern expansion of EHD to states like Michigan and other Midwestern areas, and also further up the eastern coast. And so epizootic hemorrhagic disease virus is in the family Bovidae in the genus Orbivirus. It has a double-stranded segmented RNA genome, and there are 8 proposed serotypes. The virus is vectored by Culicoides species, also called biting midges. They typically feed on sugar to survive, but females require a blood meal for their eggs. They take the protein from the blood. And currently, Culicoides sonorensis is the only confirmed vector of EHDV in America. But there are several other suspected species. So now I'm going to get to the research problem. In animal models is a living organism in which normative biology or behavior can be studied, or in which a spontaneous or induced pathological process can be investigated. So basically, what this is saying is that when you have a disease that you want to study, and you start out with your natural organism, and hopefully the goal is to move it to another animal where you can see similar disease processes or similar outcomes without having to work with the actual natural host. And so far, infection studies are typically conducted with white-tailed deer, which is the natural host, and then occasionally some cattle. But this can cause some issues because animals are not always available. We commonly get orphan baby deer or deer from deer farms. And then because especially here in the Southeast, the disease is kind of ubiquitous in the environment, they have to be housed indoors and in special units so that they don't get exposed naturally to the virus before we can infect them. And then additionally, the deer are pretty awesome to work with. There are not always reagents and kits that are commonly available for other animals available for deer. And then particularly my interest is in terms of the vector. So the only confirmed vector of EHDV is Chilocodes sonorensis. And so you can see in this picture what their general range is. And then there are some other suspected vectors which have a broader range. And then in areas where we've seen increased outbreaks, so like in Michigan, Chilocodes sonorensis is missing. And then also in some outbreak areas, there have been surveys that have showed that Chilocodes sonorensis in an outbreak area is either at very low levels or not present at all. And so this is an indicator that there are other potential vectors that should be contributing to outbreaks. And so the criteria for disease vector confirmation include recovery of a virus from a non-blood-fed well-caught specimen, demonstration of the ability to become infected experimentally, demonstration of biological transmission, and the accumulation of field evidence confirming an association of the infected arthropods and the appropriate vertebrate host. And so my goal, well, one of the goals was to be able to create a model that could satisfy at least one of these criteria, and ideally two, so that's two. So now I'm going to talk about the different models that we used. So historically, there have been murine models used for orbiviruses. They've been used to study pathogenesis, the immunological response, and we usually use newborn mice because they're immunocompromised and the infection does not occur naturally. So usually you need some type of immunocompromised state in order to get infection. And so we propose to use 2 knockout mice strains that become immunocompromised because of the absence of either the interferon alpha beta receptor deficient interferon alpha beta receptor deficient mouse or the toll-like receptor 3 deficient mouse. And so just to give you a little bit of background on both of those pathways, interferon type 1 is important in inducing an antiviral state. Loss of interferon results in suppression of the immune response, specifically the innate immune response. And double-stranded RNA happens to be a potent stimulator of the immune response. And so logically, you would think that if you removed a key component to the response of double-stranded RNA infection, you would hopefully be able to then get an infection where you would not naturally see one. And so in the INVAR negative mouse has also been used in other studies for other hemorrhagic viruses or vector-borne viruses. And this includes Congo, hemorrhagic fever virus, Ebola virus, and dengue virus. And then toll-like receptor 3 is an innate immune recognition receptor, or also commonly known as AMPs. They recognize double-stranded RNA as well, and then induce an inflammatory response and interferon beta production. And so because EHDV is a double-stranded virus, we thought if we are taking away one of the main detectors of double-stranded RNA, then we should be able to let the virus replicate more than it would in a non-immunocompromised mouse. And then our final model, or potential model, were embryonated chicken eggs. And historically, they have been used to propagate EHDV. And as Dave and the rest of the flu lab know, they also have been used to propagate flu. They have also been used in the early EHDV research for vector confidence, and then it kind of fell out of vogue for some reason. But one of the benefits is that they're really inexpensive. Eggs are about $3 a piece. They're easier to work with. They don't have to be put in a containment unit. You can just work with them inside of a hood, and you can work with a large number of them at any given time. So looking at more evidence for why these would be good models, there was an article published by Eschbaumer et al. That looked at EHDV infection in the interferon deficient mice with EHDV7. And so they took a number of 9-week-old mice and infected with either a high dose of EHDV7 or a low dose, and then also had an uninfected control. Sorry, these are out of order. And so then what they did was they isolated spleen tissue from necropsied mice and looked at the CT value. So basically looking for how much viral RNA are in these mice. And they showed that in high dose mice specifically, you saw more commonly death and also a number of clinical signs of infection. And then also high CT values, which indicates that there is a low CT values, which indicates that there's a high amount of viral RNA. And then Calvopania et al. Also published a paper where they looked at blue tongue infection in these interferon deficient mice. And so they were able to show that even with small amounts of inoculum, infar mice showed similar or basically the same clinical signs as they would in a sheep, which is the natural host for blue tongue infection, and saw large amounts of death when they infected all of their mice. And so our thought was, well, if it works for EHDV7 and it works for blue tongue, then it should also be able to work for the North American EHDVs, so 1, 2, and 6. And then Van der Sag et al. Did a paper where they evaluated a number of models for their ability to test vector confidence in embryonated chicken eggs. And so they inoculated chicken eggs and let the chilocoides feed on the eggs, and then looked to see whether or not these eggs could transfer virus to chilocoides during a blood meal. And so they then elaborated on that in another study, and were able to get in-lab transmission of blue tongue virus with wild-caught Australian chilocoides. And so we also said that we should probably test that for North American EHDVs because it would maybe help us answer some of the questions about vector confidence and what vectors are responsible for outbreaks in the US. So now I'm going to go over our specific objectives, research methods, and results. So our first objective was to evaluate the ability of alpha beta receptor deficient and toll-like receptor 3 deficient mouse strains to produce a high viremia to EHDV 1, 2, and 6. If a viremia is produced, we would determine whether infected mice can be used to infect chilocoides sonorensis through blood feeding. And then we also wanted to determine whether an infected embryonated chicken egg can transmit virus to naive midges through blood feeding. So going back to this first objective, we did a pilot study where we inoculated 6 mice with EHDV, 6 infar mice with EHDV2, and then 6 TLR3 mice with EHDV2. Mice were inoculated either subcutaneously or intraperitoneally, and then we serially euthanized 2 mice between day 2 and day 4, and then followed 2 mice out to day 14 to look for antibody development. And then once we got the results from that study, we decided to extend the study, and so we continued to inoculate 10 mice at a time of infar and TLR3 mice, doing both sub-Q and intraperitoneal inoculation, and then serially euthanizing. And then we also followed 2 mice towards day 14 to look for antibody development. And then once we had exhausted the EHDV2 inoculations, we then moved on to EHDV1 and 6, and so we took 6 infar mice for EHDV1 and then 6 infar mice for EHDV2 and repeated the process out to day 4. So we collected from each mouse after we inoculated them, and then they were serially euthanized. We necropsied and took tissue, so we took spleen, we took blood via heart puncture, lung, liver, and heart. And so tissues were placed in 1 ml transport media, homogenized and centrifuged, and then homogenate supernatant was either inoculated onto tissue culture and then also isolated for RNA And then we use quantitative RT-PCR as a backup confirmation, and then also just to see how much viral RNA was actually in each tissue sample. So the results of the pilot study basically showed that we got infection in one TLR3 mouse where we found positive virus in the spleen, and then we found 4 positive spleen samples from the INFAR mice and 1 positive lung sample, which is why we then continued to do TLR3 and INFAR for the next set of the study. However, when we progressed to the next step, we inoculated an additional 16 TLR3 mice with EHCV2 and did not get any virus isolation from any of the tissues. And so we decided that, or basically the evidence showed that TLR3 was not going to be a great model for EHCV infection. So we moved forward with the INFAR infections. And we were always able to isolate virus from spleen, which can be an indicator of secondary infection. But in addition, we were able to isolate virus from other tissues, including lung, liver, heart, and blood, which is an indicator of disseminated infection and viral replication. So we were getting virus replication in those mice. However, we were not seeing any clinical signs, and there was no deaths. And so these mice were not following what we would see in white-tailed deer, and so also really couldn't be considered a good model for disease pathogenesis disease pathogenesis. And so when we did follow those mice out to day 17, there was no virus recovered from any of the tissues that we isolated, and mice, we did run a neutralizing assay that showed mice had antibody titers of 1 to 80. And so it appears that mice are clearing the infection, developing an antibody response, and are no longer infected at least after 2 weeks. And so before we get into some of the CT, like the quantitative RT-PCR results, we did a linear regression to show the relationship between CT value and virus titer. And so there's an inverse relationship. So the higher the virus titer, the lower the CT value. And that's important to remember for the next coming slides. So here we just have the average spleen CT values by day of euthanasia. And we have our threshold value is a CT value of 35. And if you look at the EHCV2, which has the largest sample size, we see kind of a dip in CT value on day 3. And then we don't really see as much of a pattern for EHCV1 and 6, but I think it's just because we have such a smaller sample size in comparison. And then important to vector confidence, we also looked at the average CT value of blood samples. And you also see that dip on day 3 for EHCV2. And so we use that to kind of inform our process for vector feeding for the next objective, which is if a viremia is produced, then we would determine whether infected mice could be used to infect Chilocortes. And so we took these really great feeding cages that I got from Mark, and we took 1-day-old Chilocortes sonorensis lab-raised midges that we got from a USDA averager, and we shaped the belly of the mice that we were using to make sure that we could get contact between the midges and the stomach. And so I think when people think about blood feeding insects, they usually think about mosquitoes that have very long proboscis, and so this would not be an issue. But midges have very small biting mouth parts, and so if you don't, because we have a cage that kind of keeps them in, if you don't shave the hair, it's less likely that they're going to get contact with the skin, and so they won't get any blood feeding. So these are just some pictures of what insect feeding looks like for mice, and then you can see some of the blood engorged midges after feeding. They're a lot easier to see, and they're their bellies are overly full and red. And so this is kind of just the setup we used after we anesthetized the mice. So we have them resting on some tape, and then the feeding cage is on the bottom, and then these red boxes just indicate some of the bite marks, but it's a little bit easier to see in this picture here. And so in terms of midge feeding results, we used two different methods. So when we started, we were using a ketamine xylazine anesthetic, and we were not getting, and we also were shaving the mice and then using Nair to get extra hair off on the day of feeding. And so it has been known that ketamine xylazine can decrease body temperature, and so my hypothesis for why we weren't getting a large number of feeding is that the body temperature dropped too low and the mice were not as attractive to the midges, and then also the chemical smell of the Nair probably was a deterrent. And so we then tried a different method where we shaved and Naired the day before inoculation and then used isoflurane as the anesthetic instead. And we got a very much improved feeding rate. And then I wanted to point your attention to the last column where you have percent positive midges post-feeding and post-incubation period. And so we were able to get some virus positive midges after the incubation period. So to summarize this section, TLR3 mice are not a good model for EHTV pathogenesis and disease progression. INFAR negative mice develop viremia and disseminated infection. However, they do not mirror disease progression and pathogenesis in deer. And then midges do become infected after a blood meal on infected INFAR mice. So our final objective was to determine whether an infected embryonated chicken egg can transmit virus-naive midges through blood feeding. And so this is just a schematic of embryonated chicken eggs. And so because we're looking for blood feeding, we have to inoculate intravenously. And I think this is really cool. So that's why there's a slide. But basically, we intravenously inoculated a chicken egg, which is not as easy as it sounds. And so we basically have to demarcate a vein, drill a hole, and then inoculate through the hole while also simultaneously cantling the egg so you can see the vein. It was very frustrating, for sure. So we originally started looking at viral kinetics in the eggs, and so we inoculated with EHGV2, 1, and 6, and then followed them over a 4-day progression to look at virus titer over time, and we were able to indicate that best time for feeding would be between days 2 and day 3. And so from there, we inoculated some more eggs and then cut off the top of the egg so that veins are exposed. And then you take a modified 50 ml tube and you attach it to the egg, and then we put midges inside. And we placed them inside an incubator to make sure that the egg was warm and the area was dark, and then we let the midges basically go in and out of that hole to feed on the vein. And they were pretty good at it. Occasionally we had some midges that ate allantoic fluid, but we did get a good amount of blood-fed midges. And so for this, I also want to point you to the infection rate. We did have some issues with survival in terms of the midges, but the midges that did survive, we were able to get infection rates that are pretty consistent with the Vandersat et al. Paper, as well as some other midge infection trial papers. And then we were also able to get transmission not only from an infected egg to a naive midge, but then once those midges were incubated, we allowed them to feed a second time on naive, like some non-infected eggs, and we were also able to get virus transfer from those midges, those same midges, to new eggs. And so basically we were able to get an in-lab transmission cycle going between the eggs and the midges, which is pretty good and satisfies those two criteria that I was talking about earlier. We did do some blood virus titers, but they were a little bit harder to get because eggs would sometimes die before we were able to collect blood from them. But when we did, we saw inoculated BCEs with titers between 3.1, 10 times 3 to the 0.1, 10 TC50s per mil up to 10 to the 5.1, and then saw about like either the same or a 1 or 2 log difference between the donor BCE and a recipient BCE. but also just indicates that infection was transferred from donor egg to midge to recipient egg. And then sometimes we saw a really interesting pathology. And so on the right-hand side is what a normal embryo would look like. And then we started getting a lot of these dead cherry red embryos, which is a sign of virulence in the virus. It's killing them before we can get a chance to harvest them. So that was a really interesting side effect. And so to summarize that work, we were able to pinpoint a time of feeding between 48 and 72 hours for peak viremia. ECs do have viremia, and we recorded between 10 to the 3 and 10 to the 5 T-50 per mil, and we were able to complete a transmission cycle for EHTV 1 and 2, but not EHTV 6. And so once we kind of got things figured out, we also wanted to try with blue tongue virus 3. And so the interesting thing about blue tongue virus 3 is that it's not native to the U.S. Historically, but there have been multiple isolations since 1999. And then in 2006, there was an outbreak in West Virginia. In Virginia, this was the first time that BTV3 was designated the cause of death in deer in the U.S. Historically, the confirmed vector for BTV3 is Chilocoides insignis, but it's only found in the Caribbean and in parts of Florida. So we're unsure what the vector would be in Virginia or West Virginia in order to be transmitting the virus. And so we thought that this might be a good system to investigate that. But first we needed to see if we could get replication and transfer to midges with this virus. And so we did a very small study. We inoculated a few eggs and managed to get infection in 2. And so I fed midges on these as well. And we isolated 1 positive midge. And so this is very preliminary data, but I think it leads us in the right direction that we might be able to soon start using this model also for BTV3 and other exotic orbiviruses that we find in the U.S. in order to figure out which midge species are contributing to outbreaks. And so we can go over conclusions and future applications. And so basically, we evaluated different models for use in investigation of EHDV vector competence. We were able to establish full transmission cycles for ECEs and partial transmission cycle for INFAR interferon receptor deficient mice. I think future work would be testing these systems with field caught midges. So we use lab-raised midges, Chilocoides sonorensis, which is already a confirmed vector. So the next step would be to look at suspected vector species and then test them to see if they can also be infected through these models, and then whether or not they can transmit virus to naive versions within the model. I think alternative uses specifically for the interferon mice would be studies for virus-virus interactions. So co-infection studies where we look at reassortment or viral fitness of reassortments that we discover, or genetics or reverse genetics. I think it's really interesting that in nature and even in some of the background papers that we looked at for this project, blue tongue and then EHTV7 seem to cause more disease in other animals outside of white-tailed deer, and we don't really know why. And I think using something like a reverse genetics project to look to kind of take different segments and replace them with others and see if that affects virulence, or see if random mutations affect how the virus reacts both in these models and then maybe eventually white-tailed deer. And so I want to thank basically everyone at Squidus, specifically Mark Reuter, who has been really helpful to me since he got here a couple of years ago. Dave Stolnick and the lab, Clara and Nick and Becky for letting me annoy them for the last 4.5 years with silly questions. Dr. Mead and his lab, Dr. Howers and Deb, also for letting me annoy her. My committee, the Aperture, which is where we got the Chilocoides midges, specifically William Yarnell, which is who I interacted with most of the time, and then my home department, PHSI. And then on the friends and family side, I wanted to thank Brianna Williams, who's basically been my work wife for the last 4.5 years, and has taken care of me and listened to me cry on regular occasions. And then Michelle Willis, who let me crash in her apartment for the last 3 months of the summer when I didn't have anywhere to live. And then Shaquinta Richardson from Lola R. and Joan Collier, who have been my base support system while I'm here. And then Jenny Bloodgood, Caitlin Mortzliff, and Valerie Marcano, who sat through this presentation on Sunday to help me work through it. And then Jenny has read so many of my drafts at this point that she probably knows the project better than me. And then finally, I want to thank my husband, who basically lived through a 4.5-year long distance marriage so I could get a Ph.D., which is really awesome. So, question?
I'll give time to think.
Okay.
Okay. You concluded that the mouse model, the newborn mice, are really not a good pathogenesis model. And I take it that's really kind of based on the fact that of clinical science, and that makes a whole lot of sense. But as far as the virus replication, and that actually kind of mirrors what we see with white-tailed deer distribution, except blood. And could you kind of maybe?
Yeah, so I think, well, we also had issues with blood in general. So the way that we would isolate virus from blood in deer is a little bit different from what we were doing in mice. And so we had to use water to lyse the blood cells as opposed to either like sonication or like a lysis solution. And I think it's possible that the virus is not attaching to the red blood cells in the same way that they attach to deer red blood cells. So there might be some difference in the cells between a mouse and a deer, which would make sense. They're fairly different from each other.
The blood titers, that you had some CT values for blood, was that just on the positives, that were the 2 positives that you had? The titer there was...
No, it was on all of them.
It was?
Yeah, but right. And so then I think the other issue is that we were taking 100 microliters of blood, and I think it's possible that like just the dilutions between the water and the blood, we might have been like diluting it too much and kind of missing it.
Yeah, when you did PCR, did you hold blood? Did you extract the whole blood, or did you wash it?
I extracted it from the...
They were washed?
Yeah, they were washed. Mm-hmm.
Has anybody ever used, or did you attempt to use skid mice at all for your model, or do they just completely crash out and burn?
We were trying to get some, and we just never got our hands on any. So.
Right. But
I did think about skid mice, and I also thought about, like, you know how they humanize skid mice and like put human cells inside. I also thought about like, I guess, servidized.
Yeah.
Yeah. It was a thought. We just didn't get to it.
So you noted the ecology in the embryos in some of your MEC. Did you look for virus in those embryos?
Yeah. So any of the cherry red embryos, we did isolate for virus, and they were all virus positive. And usually those were the higher virus titers. So usually we got like 10 to the 5 TCIE 50 per mil from those specific embryos.
Questions?
Okay, thank you.
do you want me to stop?
Yeah.